Welcome everyone. So good morning from Boston. My name is Mayank. I'm the product manager and application scientist for protein crystallization at FORMULATRIX. And what I'm going to talk about today is how we have advanced as a company and what advancements have we made in the last 3 years in the field of high throughput protein crystallization. And I'll also talk about a few upcoming features in the software RockMaker. I'm sure that most of you, if not all, already know about RockMaker. If not, please ask questions. Okay, so to give you a brief timeline, the company was founded 16 years ago in 2002, and since then, in the last 16 years, we have come up with various products. We started off with the RockMaker, the master software, and then we came up with many imaging systems, along with Formulator, the screen builder, the drop setter, which we call the NT8, and Sonic, which is sub-micron size crystal detector. It's a very sensitive instrument and highly advanced, which you can use to identify very tiny crystals in your drops. FRAP, which is used for LCP pre-screening and is extremely useful for people who do membrane crystallization. But what I'm going to talk about today is the advancements that we have made in the field of Sonic, the multifluorescence imaging, and the pulse. It's a new instrument which we came up with this year. It's a titration flow filtration system, and I'm going to talk about that at the very end. Okay, so what is multifluorescence imaging? You know, most people use UV imaging to identify protein crystals and differentiate them between, from salt crystals. But it's not uncommon that your protein doesn't have enough tryptophan. So what do? What you can do is you bind your protein, you label your protein with a fluorescing dye, and there are many dyes available in the market. So you can choose from a variety of, fluorescein, Texas red, Alexa fluor. So you label your protein with an amine binding dye, and that enhances your signal by multifold. So as you can see in this particular example, under visible light, there are crystals hidden under the precipitate, but you do not see it. But because the protein was labeled, when you image the same drop with a fluorescein fluorescence light path, you can easily identify those tiny crystals. Similarly, the other application for multifluorescence imaging is studying protein-protein complexes. So if you have a complex of one protein and if you are working with two proteins, protein A and protein B, you mix them together, you make a complex and you set up a crystal plate, you get a crystal. You don't know for sure if that crystal is of protein A or protein B, or is it a crystal of both the proteins mixed together. So until you actually collect the diffraction data and solve the structure. So what you can do here is bind protein A with dye 1, protein B with dye 2, and then right before you set up your crystal experiment, just mix them together. Give them 5 minutes of time to bind to each other and then set up the crystal plate. Now when you get a crystal now, you image that using the first wavelength and the second wavelength, and if the crystal shows up in both, then you can be certain that the crystal contains protein A, which was bound to dye 1, and protein B, which was bound to dye 2, as is evident from this example. So here we used fluorescein and Texas red. Okay, the second part of the presentation is about Sonic. As I said before, Sonic is a highly advanced instrument used for sub-micron size crystal detection. This is a very famous example used in various email campaigns by FORMULATRIX. As you can see here, there is a sample drop which primarily looks clear, but it has some sort of light precipitate. However, when you image the same drop using Sonic, you see many microcrystals hidden under the precipitate. When you dilute the sample 200 times, you don't get enough signals, so you don't see anything in Sonic, obviously. But we made some changes to the hardware and we increased the sensitivity by 5 times. And now, as you can see, you can detect many more crystals in the same sample. And even when the sample was diluted 200 times, you still can detect fewer crystals, but you can detect crystals. So this can benefit you in 2 ways. You can either increase the sensitivity of your supersonic by keeping the exposure time the same, or you can reduce the exposure time and keep the sensitivity the same. So it's up to you. If you're a high throughput lab and exposure time is really important for you, you can obviously reduce the exposure time and keep the sensitivity the same. So to each his own. Coming on to the third part, the RockMaker software. As most of you are aware, it's the master software of FORMULATRIX, which has a complete control over your whole protein crystallization workflow. So you can go from screening your experiments to imaging them, to scoring them, analyzing them, optimizing potential hits, and repeating the whole process until you finally get a crystal. And once you do, you can generate various kinds of reports, share it with your collaborators, with your colleagues, with your bosses, and the scientific community. The two major features, and as you all know, we keep coming up with major updates to RockMaker software at least twice a year. But the two big features we are coming up with by the end of January next year are crystal tracking and machine scoring. So crystal tracking is a feature that would be used to document your crystals when they go on a synchrotron trip. When you get a crystal, you freeze it and then you store it in a certain pin position of a puck and in a doer, and then you send it to a synchrotron trip. Usually most labs, they have some sort of Excel report or handwritten notes on keeping a track of which crystal went to which trip, in which doer and in which puck. Now you can do all that in RockMaker. So I'm going to show you a few mock-ups that we have prepared. It's still not implemented entirely, but at least you'll get an idea of how this whole workflow is going to work. So the first thing you have to do is define all the pucks and all the doers in your RockMaker software. And obviously this only needs to be done once, unless you buy a new puck or a new doer. And once you have defined all those pucks and doers, you will define your upcoming synchrotron trip. So let's say you have a trip coming up in December, you define that trip. And then you start navigating through various wells in your various experiments. And as you come across a crystal, you would click on this Harvest Crystal button and draw a box around a certain crystal in the drop. When you do that, a window will pop up, and here you'll enter all the data that's needed. So for example, you'll assign this particular crystal to a trip, to a doer, to a puck, and to a position in that puck. Now all these fields are linked to each other. So the software will know that puck 1 is a part of doer 1, and this doer 1 is going to go to first trip. You can also define other things such as cryoprotectant, soaking. So if you have any information that you want to, you can add here. It's not mandatory, but it's good for record keeping. And see this field here? So this field is going to be auto-populated based on what you enter in the CSV file that you create later on. So I'm going to get to that in a second. So once you are done with assigning multiple crystals to multiple positions and multiple pucks, you can create a synchrotron trip report by clicking on this button. And this report can be generated as a CSV file or a much cleaner PDF file. But the real workflow is you'll click on this button, you'll create a CSV file, you'll print the CSV file, take it to your lab, and now really start harvesting crystals from your wells. Now, as you store crystals in these particular positions in pucks and doers, obviously mistakes are bound to happen. So let's say you drop crystal 3 or crystal 3 breaks as soon as you dip it in cryoprotectant. So the whole numbering system is going to change, right? So position 3 is not going to contain crystal 3 anymore. It's going to contain crystal 4. So nothing to worry. You have your printed report. Just make changes to that report by hand. And then when you have time, come back to your lab, come back to your computer, make changes, fare up those changes in this CSV file. And then you can upload this CSV file back into your RockMaker. And RockMaker will take care of updating the whole database again. Now, these other fields that you have here, these need to be filled when you're actually collecting data. So when you're ready to send your crystals to the trip, you'll send this CSV file as a printout, to the facility. And then while the data is being collected, you can enter these fields here. So as you fill these fields, and when you solve the structure, you'll enter a certain resolution of the structure and a link to the PDB, either the PDB or to the local depository that you have in your lab. And then you are supposed to upload this file again into RockMaker. So RockMaker will then fill up all these fields that I showed you, here. So basically, we are trying to make RockMaker evolve into an electronic bookkeeping system. You will be done with using your notebooks and Excel files. All the data is going to be in one single place in RockMaker. So if a lab member leaves, the PI would know exactly where to find a certain, a certain file or data related to a certain crystal. The other feature that we are going to implement in RockMaker in January 2019 is Marco, and Janet Newman from Cicero is going to talk about that in the second session today. So if you are interested, please register yourself for the second session and attend her talk. So Marco is an autoscoring algorithm developed by a Google employee, and it classifies your drops under 4 categories. It tells you the probability of a drop being clear, precipitate, crystal, or if it cannot fit it into any of these categories, it classifies it as other. However, this only works with visible light path for now. Okay, now the final part of this presentation, I'm going to talk about Pulse. It's a tangential flow filtration system. So it is something that you would use to purify, after you're done with purification of proteins, this is what you'll use to concentrate your proteins down to a certain level, or to buffer exchange it with a buffer of your choice. So most people have been using, centrifuges for this purpose, which is fine, but it requires a lot of human intervention. So this provides you with a true walkaway solution. All you have to do is load your Falcon tubes right here, and then program customize your cycle, the protocol, and then just walk away. You can always monitor the progress of your concentration cycles in your smartphone. Now I'm going to show you a very quick video of how this Pulse looks like. It's a very tiny footprint, and this has only come out this year, so it's still we've started taking orders, but it will be in full flow next year. So this is how you would load your 2 Falcon tubes, and we can take from 50 ml to 100 ml solutions. This is the membrane that fits on the top of the Falcon tubes, and this is the LED screen, which based on Raspberry Pi basically, and you can program your protocol cycles using the UI here. So the protocol is that the stuff from the protein sample from the Falcon tube gets aspirated into this and flows through this membrane, and it gets filtered out. And of course, these membranes are available in different sizes, different cutoffs. So it will get filtered off based on a tangential flow filtration system, which means that you are passing your protein in a horizontal direction while it's being filtered in a vertical direction. So this helps in your protein not clogging your membrane. And as the protein gets concentrated, it dispenses back into the same Falcon tube. So you can either use the second Falcon tube as a second protein solution or as a new buffer. And whatever is filtered out gets collected in the Falcon tubes, while the reservoir placed on the right side collects all the waste that you can throw off later. So with that, I would like to conclude my talk. I hope we are running on time. If we have any time for questions, I would like to take it up. Otherwise, we can always email you the answers back.